good morning, everyone. My name is Mónica Carvajal. I'm a scientist working at the Genetic Resource Program of the Alliance Bioversity International and CIAT, which is based in Cali, Colombia. And I'm going to give present a case study on the use of DSI to compare international cassava collections, which are hosted by CIAT and by IITA. So this is a work that has been jointly performed with IITA. So I think Carolina and Marie-Noël has already explained that DSI help us shed some light on the genetic relationships among the accessions that are conserved in the gene banks. And this is important because it facilitates germplasm curation and also enhance the use of diversity for crop improvement. So cassava is an important tropical root crop because its starchy roots are a major source of dietary energy for more than 500 million people in the tropics. So most of the crop diversity is conserved in 2 international gene banks. So one comprises almost 6,000 accessions and is conserved at the Alliance Bioversity and CIAT. And the other collection is conserved by IITA in Africa, and it is comprised mostly by a bit over 4,000 accessions. So the Alliance collection represents the diversity of the center of domestication in Latin America. And as you can see in the pie chart, it's mostly composed by land races, but we also conserve some improved varieties, which are product of the breeding programs and cassava wild relatives. So since 2016 and from 2016 to 2021, so we have been collecting tissue and extracted DNA from each of the plantlets accessions that we conserve. So we conserve cassava in vitro. And we have used DART-seq as genotyping platform to produce the DSI for the cassava collection. So we have managed to complete 97% of the collection being genotyped. So with DSI, as it has been already shown, allow us to perform different kind of studies. We wanted to first have a view of what is the genetic diversity, how is the diversity of the collection that we conserve. So Carolina already explained, so this is a diagram that represents the genetic diversity among accessions. So each dot represents an accession. And as you can, the closer the dots are, the more similar they are, and the separated they are, the more distant. So you can clearly see a big cloud in the middle that represents the cultivated land races and improved varieties of cassava. And there are some more distant groups represented in green color, which are the wild relatives that are, of course, farther apart from the cultivated cassava, but we see some green dots that are closer, which represent those subspecies of cassava that contributed or are closely related to the cultivated version. So taking advantage that IITA has also genotype or produced DSI information from their collection using the same genotyping platform. So we thought it would be very interesting to study the 2 collections together. So these pie charts show how is the composition of the IITA collection. So they conserve in 2 sites cassava, but you can see that the majority represents breeding materials and some land races. So we, in 2022, so we co-analyze or put the 2 DSI of the 2 collections together, and we perform a joint diversity analysis. As you can see in the table, you can see how was the composition of the samples that were included in this analysis. So it was very interesting. As you can see in the diagram, orange dots represent the accessions conserved by IITA, and green dots represents the accession conserved by us in CIAT. So you can see that there is a clear complementarity of the clear genetic complementarity between the 2 collections. So this is in addition to being very nice, that the two collections are complementarity, and they have complementarity. There is a bit of genetic redundancy across the two collections. So this is a good example of how DSI can also aid conservation management, can support conservation management and facilitate the access and use of crop diversity. So with this study, we are producing a knowledge base for a coordinated stratified curation of the two collections. So we have been working on identifying the genetic duplicates or the genetic redundance within and across collections. This is important because we can optimize the conservation costs. Conserving plants in vitro is costly. It also is important to identify those possible redundant accessions, because if there is any germplasm evaluation or characterization, so we can optimize the resources invest for that. It's also helping us to guide the efforts that we want to invest for cryopreservation of the cassava collection, as mentioned as well by the other colleagues. So we can also use this information to identify gaps in the collection. And we are in the process of redefining new core collections within and across the two gene banks. So Carolina also gave an example of how we can make more use and support crop improvement with the DSI that we are generating. And we are also participating in this multi-center project, which is led by CIMIT and funded by Bill and Melinda Gates Foundation. So we are working together with IITA, leveraging the existing DSI and mixing this information with the bioclimatic variables that are extracted using the geolocation data of collection sites of the accessions. And we are performing, as Carolina explained, the environmental association analysis. So we have identified some possible candidates, allele that are candidates that are under revision. And so I think it's important to also have perspective on how this work can help us in the collaboration with other gene banks. So we think that this is the way to start exploring the collection. So we have been in contact also with ILRI, which also conserve tropical forages as we do. We have some common species that we conserve. We both agree to use the same genome-wide genotyping platform, and we are going to work in a similar way as we did with IITA to understand the Brachiaria, also known as Urochloa, and the Panicum, known as well as Megapyrsus, the collections that the two institutions conserve. So with that, I finish, and thank you. So this is some of the people that contributed with this work and funding institutions. Thank you.